Okay, let's dive right in. So the really striking thing, maybe the most counterintuitive finding from today's sources, is this. If you block a specific off switch in key immune cells, these are called dendritic cells, specifically by targeting a protein called STAT3, you can actually seriously boost how well cancer immunotherapy works.
Yeah, even in cases where the standard stuff fails.
Exactly. It seems like blocking this STAT3 break lets another protein, STAT5, kind of take over and supercharge these dendritic cells. Makes them better tumor fighters.
That's right. And this really interesting research, it was just published in Nature, May 14th, 2025. It takes a close look at this critical balance, really, between two signaling pathways inside those dendritic cells.
Okay.
And this balance, this sort of push and pull, is turning out to be really key for understanding how our immune system fights cancer, and crucially, why these newer therapies, anti-PD-1, anti-CTLA-4, why they work great for some people but not others.
Right. So just a little background here for everyone. Cancer immunotherapy, it often really depends on these dendritic cells. Let's call them DCs.
Yeah, DCs is easier.
Think of them as like the intelligence agents of your immune system. They spot bits of the tumor antigens and show them to the killer T cells.
The soldiers, basically.
Yeah, the soldiers who then go attack the cancer. But the area right around a tumor, the microenvironment, it can send out signals that kind of tell these DCs to slow down, to be less effective.
Makes them sluggish.
And inside these DCs, you've got these two proteins, STAT3 and STAT5, playing opposite roles. STAT3 generally dampens things down, suppresses immune activity.
Right, the off switch you mentioned.
While STAT5 does the opposite. It promotes immune activity, helps the DCs mature, get ready to fight.
Yeah, they on-switch in a way.
And what researchers have noticed is that tumors often manage to activate STAT3, which makes the DCs dysfunctional, and then the immunotherapy, the ICB treatments, they just don't work as well.
Exactly. So this new research focuses right on that ratio, the STAT5 activity versus the STAT3 activity inside the DCs. The idea is that this ratio is, well, vital for a good anti-tumor response and for ICB to succeed.
So in this deep dive, we're going to unpack how tweaking this balance could maybe lead to some new cancer therapies.
Okay, so the study, it kicks off by looking at data from actual human cancer patients, people getting immune checkpoint blockade, ICB, that's figure 1.
Okay.
They analyzed RNA sequencing data that tells you which genes are switched on or off in the cells, and they looked at it in bulk, like all cells mixed together, but also cell by cell, which is single cell or cRNA-seq.
Wow, okay. Across different patient groups.
Right, 3 different cohorts. The first was from the University of Michigan, patients getting various ICBs. They looked at how well they responded, complete response, partial, stable disease, or if the disease progressed.
And how long they survived, presumably.
Exactly. And they tried to link that response and survival to what was happening immunologically.
And what did they find? Any immediate patterns?
Yeah, they did. They saw that patients who had higher activity scores for their CD8 plus effector T cells, those killer T cells we talked about, and also higher scores showing that a specific type of DC, called DC1, was mature and active. Well, those patients tended to live longer.
Okay, that makes sense. Active killers, active messengers.
Right. And what was really interesting was the strong link between the two. More mature DC1s seem to go hand in hand with more active killer T cells.
Okay, so they're connected.
Very much so. But then they dug deeper into the STAT5 and STAT3 gene activity within those DCs.
The core idea.
Exactly. And here's the kicker. Patients who had both high DC1 maturation scores and a high ratio of STAT5 gene activity compared to STAT3 gene activity. They had the best overall survival, the longest.
Wow. So it's not just having the cells, it's this specific internal balance within the DCs.
Precisely. And they saw similar results when they looked not just at the genes, but at the whole STAT5 signaling pathway activity relative to STAT3 expression. The balance seems critical.
Okay, one cohort, did they check this elsewhere?
They did. They looked at a second group, melanoma patients. Melanoma often responds pretty well to anti-PD-1 CTLA-4.
Right.
And yep, same pattern. High DC1 maturation plus that high STAT5, STAT3 ratio correlated with the longest survival there too.
Okay, consistency. That's good.
And the third cohort gave even more detail. This was triple-negative breast cancer patients. And here they had single-cell RNA-seq data from before and after ICB treatment.
Oh, that's powerful. You can see the change.
Exactly. And in the patients who responded to the immunotherapy, after treatment, their DCs showed increased STAT5 signaling and decreased STAT3 signaling.
The balance shifted.
It shifted, but in the non-responders, no such shift. Their STAT5, STAT3 balance didn't really change.
Okay, that's pretty convincing.
And they drilled down even further, looking specifically at conventional DCs or CDCs. In the responders, post-treatment, you saw more STAT5, less STAT3 compared to before treatment and compared to the non-responders after treatment.
So the big takeaway from Figure 1 seems to be that successful ICB treatment is linked to, and maybe even helps cause, this favorable shift in the STAT5, STAT3 balance inside DCs. Especially these CDCs. It pushes towards the immune-boosting STAT5.
You got it. That correlation in humans is strong. So naturally, the next question is how? What's the mechanism? And that's what Figure 2 gets into, using lab models.
Right, moving from correlation to causation, or at least mechanism. What did they do in the lab?
They used both cells in dishes in vitro and also experiments in mice in vivo.
Okay.
First, in vitro, they used a mouse DC cell line called Jiajia. And here's a neat finding. When they use a technique to knock down or reduce the STAT3 gene.
Less STAT3 protein.
Right. They saw an increase in the activation, the phosphorylation of STAT5.
So taking away STAT3 boosted STAT5 suggests STAT3 normally holds it back.
Exactly. It acts like a restraint. They also looked at the pathway that activates STAT5, involving something called GM-CSF and its receptor, GMR, and an enzyme, JK2. Knocking down parts of that pathway reduced STAT5 activation, as expected.
So STAT5's activation runs through GMR and JK2.
Correct. And it looks like STAT3 is somehow messing with that whole GMR, JK2, STAT5 axis.
Interesting. How do they confirm this in a more, complex setting, like in mice?
Good question. They used genetically engineered mice where the STAT3 gene was deleted specifically in CDC1s, that key DC subtype.
So only the CDC1s lack STAT3.
Precisely. And guess what? In the mice, those CDC1s without STAT3 showed more activated STAT5 compared to normal CDC1s.
Okay, consistent with the cell line data.
Yep. And they used a broader screen, a phosphoreceptor kinase array, which confirmed higher activated STAT5 in those knockout CDC1s. But not just that the STAT3 knockout CDC1s also showed higher levels of those maturation markers.
MHC-I, MHC2, CD80, CD86.
Yeah.
The stuff DCs need to activate T cells.
Exactly. They looked more prepared, more mature.
So how does STAT3 actually interfere? Did they figure that out?
They got some clues. Using co-immunoprecipitation, a technique to see if proteins stick together, they found that losing STAT3 actually increased the interaction between GMR and STAT5 and also between JK2 and STAT5.
So maybe STAT3 normally gets in the way, physically prevents those interactions that activate STAT5.
That seems to be the idea. And to show STAT5 was really driving the maturation, they inhibited STAT5 in the STAT3 knockout cells and saw that the levels of MHC-II and another immune signal, IL-12, went down.
So STAT5 is definitely needed for those functions.
Definitely. And finally, looking at the RNA again in these STAT3 knockout CDC1s, they saw whole sets of genes related to maturation, antigen processing, presentation, IL-12 production were all upregulated, more active.
Wow.
And using another technique, ChIP-PQ-PCR, they showed STAT5 itself was binding more strongly near the genes for those maturation markers in the knockout cells.
Okay, so Figure 2 really nails down the mechanism. STAT3 holds back STAT5 signaling, probably by interfering with the GMR-JK2 pathway, and this limits DC maturation and function. Removing STAT3 unleashes STAT5. That's the core mechanical insight, yeah.
So if removing STAT3 makes DCs more mature and active in the lab, does that actually translate to better anti-tumor immunity? That seems like the next logical step. Figure 3. Precisely. Figure 3 looks at the functional impact. First, another in vitro test. They took the STAT3 knockout CDC1s and control CDC1s, loaded them up with a known tumor antigen.
A piece of the tumor for the T cells to recognize.
Right, the SINFEKL peptide in this case. And then they mixed them with specific T cells, OTI T cells, that recognize that antigen.
And what happened?
The STAT3 knockout CDC1s were way better at firing up those T cells. They saw more T cell proliferation, more production of those key immune signals like IFNO and TNF, and more granzyme B.
Granzyme B, that's the stuff T cells use to actually kill tumor cells, right?
That's the one. So better T cell activation in the dish.
Okay, promising. But does it work against actual tumors in mice?
That was the big test. They used mouse tumor models, MC38 colon cancer and B16F10 melanoma. They put these tumors into the mice lacking STAT3 in their CDC1s and into control mice. And the tumors grew significantly slower in the mice whose CDC1s lacked STAT3.
That's a real anti-tumor effect.
Definitely. And when they looked inside those slower growing tumors, the STAT3 knockout CDC1s that were there showed, again, higher STAT5 activation and more MHCII and CD86.
Just like in the earlier experiments.
And importantly, in the tumors in the nearby lymph nodes of these mice, they found more of those beneficial activated CD8 plus T cells, the ones making IF-NA and granzyme B.
So figure 3 connects the dots. Removing STAT3 from CDC1s boosts their ability to activate killer T cells, and this leads to better control of tumor growth in vivo. STAT3 in DCs really does seem to suppress anti-tumor immunity.
It strongly supports that conclusion.
Which naturally leads to figure 4.
Yeah.
Can we actually do this therapeutically? Can we drug STAT3?
Exactly. This is where it gets really exciting from a potential treatment perspective. They started looking at pharmacological ways to target STAT3, specifically using molecules called PROTACs.
PROTACs. I've heard of those. They don't just block a protein. They actually get the cell to destroy it, right?
Yeah, precisely. They're like targeted protein degraders. They developed 2 STAT3 targeting PROTACs called ST36 and later an improved one, ST2301.
Okay, so did ST36 work on DCs in the lab?
It did. In vitro, they showed ST36 got rid of STAT3 protein in mouse CDC1s, and the more drug they added, the more STAT3 disappeared dose-dependent.
And did degrading STAT3 have the same effect as knocking out the gene? Did it boost STAT5?
Yes. Treating normal CDC1s with ST36 enhanced STAT5 activation, but crucially, it didn't boost STAT5 in the CDC1s that already lacked STAT3, which makes sense.
Confirmed STAT3 was the target, holding STAT5 back.
Exactly. And just like the genetic knockout, ST36 treatment increased the interaction between GMRJ2 and STAT5, and it led to higher expression of those DC maturation markers.
MHC, CD80, CD86.
All went up. And they checked it wasn't some off-target effect via, say, STAT1 or another STAT protein. Knocking down STAT1 didn't stop ST36 from boosting maturation. It really seems specific to degrading STAT3 and unleashing STAT5.
Okay, the PROTAC works as expected in vitro. What about in tumor-bearing mice? That's the real test.
And the results were pretty striking. ST36, given just by itself, showed significant anti-tumor effects in several different mouse tumor models.
Which ones?
They tested it in models of breast cancer, 4T1, colon cancer, MC38, ovarian cancer, ID8, and even Lewis lung carcinoma, the LLC model.
LLC. Isn't that one known to be pretty resistant to standard immunotherapy, like anti-PD-1?
It is, so showing efficacy there as a single agent was quite impressive.
Wow. And did it work on bigger tumors too, or just early ones?
They showed it was effective against established large tumors, and it even reduced metastasis, the spread of cancer.
That's huge. So Figure 4 shows that pharmacologically degrading STAT3 with a PROTAC like ST36 can enhance DC function and actually shrink tumors, even resistant ones, all on its own.
Yeah, a really significant finding.
Okay, so ST36 works. Figure 5, then, I assume, digs into how it's working in the mice and whether it can be combined with other treatments.
Spot on. Figure 5 focuses on the mechanism of action in vivo and synergy with ICB. First, they showed the anti-tumor effect of ST36 completely disappeared in mice with no immune system.
Like NSG mice or RAG knockouts.
Exactly. Which tells you it's working through the immune system, not by directly killing tumor cells.
Makes sense. An immune mechanism.
Then they depleted specific immune cells, get rid of CD8 plus T cells. The effect of ST36 vanished.
So it definitely needs those killer T cells.
Absolutely. And consistent with that, ST36 treatment led to more of those active cytokine-producing CD8 plus T cells inside the tumors.
Okay, so it boosts the T cell attack.
Yeah.
Does it depend on the DCs, specifically the CDC1s?
Crucial question. They tested it in BATF3 knockout mice, which naturally lack CDC1s. And in those mice, ST36 had no anti-tumor effect.
Wow. So it absolutely requires those CDC1s to work.
It does. And they confirmed ST36 was actually getting into the tumor, degrading STAT3 in the DCs within the tumor, reducing activated STAT3, increasing activated STAT5, and boosting those maturation markers on the tumor DCs.
So it's doing exactly what they predicted based on the earlier work.
And just to be absolutely sure STAT3 was the target, they showed ST36 didn't work in mice that already had STAT3 knocked out in their DCs. You can't degrade a protein that isn't there.
Right, the ultimate confirmation.
They even did these cool adoptive transfer experiments. They took mice lacking CDC1s and gave them back either normal CDC1s or STAT3 knockout CDC1s. ST36 only worked if the mice got the normal CDC1s, the ones that still had STAT3 for the drug to target.
Okay, that really pins it down to degrading STAT3 in the CDC1s.
And they did similar experiments showing STAT5 in the CDC1s was also required for ST36 to work. So it's degrade STAT3, unleash STAT5, activate DCs, activate T cells, kill tumor.
A clear chain of events. Now, what about combining it with existing therapies, like ICB?
This is where it gets potentially very impactful. They combined ST36 with an anti-PD-L1 antibody, a standard checkpoint inhibitor. Profound synergistic effects. Much better tumor control than either agent alone in both MC38 and B16F10 models.
Synergy. That's what you hope for,
Yep.
making existing therapies work better.
Exactly. And they also tested their newer, more potent degrader, SD2301. It also showed significant activity by itself and synergized really well with anti-PD-L1.
And ST2301 also worked via STAT3 and CDC ones.
Yep, same mechanism.
Yeah.
Dependent on STAT3 and DCs and the presence of CDC ones.
So Figure 5 wraps it up nicely. These STAT3 degraders work by hitting STAT3 and DCs, boosting STAT5, improving DC function, driving a potent T-cell response, and they look really promising in combination with ICB.
That's the story, yes. A very compelling one.
This really does feel like a potential breakthrough. What would you say are the biggest innovations here? What makes this research stand out?
Well, there are several things. First, the conceptual advance really defining the STAT5-STAT3 balance as a key control point for DC function in the tumor microenvironment and for ICB response. It refines how we think about immune suppression.
Okay, a clearer understanding.
Second, the technology application. This is really the first time STAT3 PROTAC degraders have been successfully used like this to tweak innate immune cells, the DCs, in vivo for cancer therapy. Most PROTAC work before was aimed right at the tumor cells.
So a new way to use PROTACs in immuno-oncology.
Exactly. Third, the development of these novel drugs themselves, SD36 and especially SD2301. They're potent, selective, and they work in tough preclinical models, including ICB-resistant ones. SD2301 looks like a serious candidate for clinical development.
Right, the improved potency and properties.
Fourth, the detailed mechanism. They really dissected how STAT3 inhibits that GM-CSF-JK2 STAT5 pathway, giving a solid molecular reason why targeting STAT3 should work.
Understanding the why is crucial.
And finally, demonstrating that synergy with ICB. That's huge, because overcoming resistance to current therapies is a major goal.
So where does this lead? What are the next steps, future directions?
Well, the immediate implication is that these STAT3 degraders could be a whole new class of immunotherapy drug, particularly perhaps for patients whose tumors don't respond well to current ICB or those with advanced metastatic disease.
A potential new option.
Absolutely. But of course, moving to humans requires care. They'll need further studies on the selectivity and safety of these PROTACs in human DCs.
Preclinical safety testing.
Right. But clinical trials seem warranted, based on this data, to test these degraders, maybe SD2301 first, both alone and combined with ICB in cancer patients. The fact that the university has patented and licensed the tech suggests they're serious about clinical translation.
That's a good sign.
Eh?
Anything else this opens up?
Yeah, it makes you wonder about other signaling pathways in DCs. Are there other balances, other targets we could hit to boost anti-tumor immunity even more? And also, could STAT3 degraders be combined with other types of immunotherapy beyond just checkpoint inhibitors? Maybe vaccines, HAR T cells, lots of possibilities to explore.
So summing it all up, the big takeaway is, by understanding this intricate STAT5, STAT3 balancing act inside dendritic cells, and then cleverly using these PROTAC degraders to specifically remove STAT3, this research offers a really promising new way to empower our immune system against cancer. It could improve outcomes, especially for hard-to-treat cases.
Absolutely. It's a fantastic example of how digging deep into fundamental cell biology, into these signaling pathways, can unlock genuinely innovative therapeutic strategies.
It really makes you think. Given how complex immune cells are, with all their internal wiring, what other yin and yang relationships these kinds of opposing forces might be hiding in there? Could we find other switches to flip, other dials to tune to make our immune responses even better against not just cancer, but maybe other diseases too?
That's the exciting question, isn't it? This work certainly suggests there's a lot more to discover by carefully dissecting these fundamental processes.